Welcome back, everyone. Today, our topic is tissue culture, which is one more subunit of your third unit. Okay, and in tissue culture, we are mainly concerned to study what tissue culture means, what is cellular totipotency, what are the different techniques of tissue culture, and what are the applications of tissue culture. First of all, in this part, we are going to introduce ourselves to the field of tissue culture, and we will also study in brief the concept of cellular totipotency. So let's move on with the concept of tissue culture. The plant cell and tissue culture is a technique of growing cells, tissues, or organs in an artificial sterilized nutrient medium under aseptic controlled conditions. If you are culturing the cells, tissues, or organs of plants in culture medium, okay, you are having a culture medium, nutrient medium, and you are culturing these plant parts under aseptic conditions, that means under sterilized conditions, and you are having controlled temperature, you are having controlled light, you are doing tissue culture. Okay, the plant materials that you culture in the cultural laboratories may include cells, which I have already told you, tissues, or organs. If you are culturing organs, these may include the excised root tips, shoot bud, leaf petiole, anther. Okay, anther means the upper part of the stamen, which contains the pollen grains. Or you can also culture the embryo, ovule, or at times in plant tissue culture laboratories you can culture the ovary as well. Okay, I will repeat, the plant materials that you wish to culture, it may include the cells, tissues, or organs, such as you have excised root tips, shoot buds, leaf petiole, anther, embryo, or you can at times culture the ovules or even ovaries. Okay, now there is one more term, which is explant. Explant is simply the plant part that you have cut from a plant, and now you are going to culture this plant part in the laboratory, in the culture medium. Okay, explant means that plant part which you are culturing in the culture medium. Here you can see in this image a number of different plant organs are being cultured in the culture laboratory using the petri plates and test tubes. Okay, now the concept of cellular totipotency. The concept of cellular totipotency, it is so much important that the whole subject of tissue culture is based on it. Okay, you will come to know and we will discuss it. First of all, the term cellular totipotency, it was used for the first time by a German botanist, Gottlieb Haberland, in 1902. He gave an idea that every plant cell is totipotent. By totipotent, we mean the plant cell is capable of giving rise to an entire new plant. If a plant cell is capable of giving rise to an entire new plant under culture conditions, that plant cell is known as totipotent cell. Okay, so if you have to define totipotency, it is capability or capacity of a cell to give rise to a complete plant when cultured in a suitable culture medium at appropriate temperature and aeration conditions. Okay, you are taking a plant cell and now you are putting it on a petri plate, and now the petri plate is also containing the culture medium. Okay, and you place this culture medium in the culture laboratory where you have the controlled conditions of temperature, you have controlled aeration conditions. If this cell is capable of giving rise to the whole new plant, that cell is known as totipotent cell. Okay, and the totipotency is because of two unique properties of plant cells. One is known as differentiation and second one is known as redifferentiation. First of all, let's understand the concept of differentiation. Differentiation is actually the transformation of mature and differentiated cells to meristematic state, leading to callus formation. Okay, अगर आप एक काम करोगे, you will have a number of differentiated cells from the plants. You will cut down a plant pot, okay, and that plant pot contains only differentiated cells. That means they are not meristematic. They are not capable of division. You are taking these cells from the plant and you place these cells in a nutrient medium. These cells will give rise to a mass of cells which is known as callus. That means the differentiated cells have now retained their power of division. This property of cells by which they can transform from differentiated state to the meristematic state, this is known as differentiation. ठीक है बेटा? This is known as differentiation. Now another one is redifferentiation. Now you are having a callus tissue, okay? The ability of the dividing cells of the callus, now you have a number of cells in the callus. Callus is constituted of a number of cells. The ability of the dividing cells of the callus to form entire plants in presence of proper nutrients and hormones. Now you will take a few cells from this callus and you will place these cells in, or you will take a single cell and you will place this cell in a nutrient medium containing proper hormones and nutrients which are suitable for the formation of the plant. If this cell gives rise to the whole plant again, that means the meristematic cell, the dividing cell, is giving rise to the whole new plant. This process is known as redifferentiation. Now the dedifferentiated cell is getting redifferentiated. It becomes differentiated again. Okay. This differentiation and redifferentiation, it forms the basis of tissue culture. It forms the basis of plant tissue culture. Okay. That means all the plant tissue culture experiments are dependent. They are based on the concept of dedifferentiation and redifferentiation. Or you can simply say the whole tissue culture, the whole subject of tissue culture is based on the concept of cellular totipotency. Okay, this is all about the cellular totipotency and dedifferentiation and redifferentiation. In the next part, we will start with the techniques of tissue culture. Till then, stay blessed and thank you.